There is a company in the Woodlands, Texas that most Americans have never heard of. It does not have a storefront. It does not have a consumer product. It does not advertise. It does not have a headquarters that looks like anything other than a working scientific laboratory, because that is exactly what it is. Its name is Othram, and if you follow true crime with any depth, you have heard that name in the last 5 years. Not because Othram promotes itself, but because Othram keeps showing up at the end of stories that did not have endings. The Golden State Killer had an ending. The Long Island serial killer had an ending. 40-year-old Jane Does who had been buried without names had endings. Cold cases that local sheriff's offices had closed, reopened, and closed again across multiple decades of failed investigation had endings. Othram gave those cases endings. And as of the confirmed reporting on the Nancy Guthrie investigation, Othram, or the specific forensic genealogy pipeline that Othram represents, is now involved in this case. Tonight, that is the story we are telling, not the evidence you already know, not the glove, not the porch blood, not the backpack. Tonight we go inside the laboratory, inside the specific science that closed the Long Island serial killer case after more than a decade of failure, inside the method, inside the machine, inside the thing that transforms a hair sample, a degraded DNA fragment, or a cold biological clue into a name on a warrant. Because if that same pipeline is now working the Nancy Guthrie evidence, then the public understanding of where this investigation stands is significantly behind where the science actually is. And by the end of this video, you are going to understand exactly why. Let's start with Lisk, the Long Island serial killer. Because that case is the closest structural parallel to the Nancy Guthrie investigation that exists in the recent public record, and understanding how Othram cracked it tells you precisely what the lab is capable of doing with the evidence that exists in this case. The Long Island serial killer case began, for most of the public, in December 2010. That was when investigators searching for a missing woman named Shannon Gilbert walked the barrier beaches of Gilgo Beach on Long Island and found something that stopped the search entirely. Not Shannon Gilbert. 4 sets of human remains, partially skeletal, wrapped in burlap, laid out along Ocean Parkway in a pattern that was unmistakably deliberate. Shannon Gilbert's body was found separately, in a marsh, more than a year later. But the 4 Gilgo Beach victims, later joined by others as the search expanded, became the focus of one of the most publicized cold case investigations in the northeastern United States. The burlap, the barrier beach, the deliberate spacing, the victims all linked through their digital footprint to online escort advertising. A killer who had been operating with apparent impunity for years, possibly decades, along a stretch of Long Island highway that locals drove daily without knowing what had been placed in the brush beside the road. For more than 10 years, the case had a profile and no suspect. Law enforcement investigated, tips came in, persons of interest were identified, examined, and cleared. The case remained open, remained national, and remained without a name attached to it. And then, in 2023, a man named Rex Heuermann was arrested, a Manhattan architect, a Massapequa Park homeowner, a person who had lived and worked in the geographic footprint of the investigation for decades without ever being publicly identified as a suspect. He was charged with 6 murders. He has since pleaded guilty to 6 counts and is awaiting trial or sentencing on additional charges. The Long Island serial killer, a case that spent more than a decade as one of the most stubborn, unsolved serial murder investigations in the country, had a name. And the DNA technology that connected Rex Heuermann to those victims, that built the biological bridge from the evidence collected on those barrier beaches to the man who lived 45 minutes away, was the same generation of forensic genomic technology that is now working the Nancy Guthrie evidence. Forensic Investigative Genetic Genealogy, or IgG, is the methodology that made the difference in LISK, and that represents the most significant advance in the forensic identification of unknown suspects since the introduction of DNA testing itself. Here's how it works, from the ground up. Standard forensic DNA analysis, the kind that has been used since the late 1980s and that the FBI's National DNA Database, SOTUS, relies on, generates a profile by examining specific predetermined locations in the genome called short tandem repeats, or STRs. An STR profile is essentially a forensic fingerprint, a numerical pattern derived from 13 to 20 locations in the genome that is statistically unique to each individual, except identical twins. The power of STR profiling is in its simplicity and its database. CODIS contains more than 20 million STR profiles, offenders who have been convicted of qualifying crimes, arrestees in jurisdictions that require collection at arrest, and voluntary submissions. When you upload an unknown STR profile from a crime scene and it matches a profile in CODIS, you have a direct identification. Name, date of birth, criminal history, and a court-admissible result. The limitation of STR profiling is also in its database. If the person who left the evidence at the crime scene has never been arrested, never been convicted, never submitted voluntarily to DNA collection, and has no relatives in the database who share enough genetic material to flag a familial match, then CODIS returns nothing. The profile is unknown. The database has no answer. The investigation stalls. That is exactly what happened with the glove DNA in the Nancy Guthrie case. An unknown male STR profile, submitted to CODIS, no public match returned. Investigation stalled on that specific evidentiary pathway. IgG is what happens when CODIS cannot answer. Instead of examining 13 to 20 predetermined STR locations, IgG sequences the entire genome, or as much of it as can be recovered from the available biological material, using next-generation sequencing technology. This produces not a profile of 20 data points, but a map of hundreds of thousands of genetic variants spread across the full 3 billion base pair human genome. That map is not compared against CODIS. It is compared against consumer genealogy databases, GED match, family tree DNA, where millions of people have uploaded their own genetic profiles for ancestry and family history purposes. People who have never been arrested, people who have no criminal record, people who took a 23andMe test or an ancestry DNA kit because they wanted to know if they were Irish, or because a cousin told them the family tree was worth exploring, or because they were adopted and wanted to find their biological relatives. When the unknown perpetrator's genomic profile is uploaded to those databases, it does not need to match the perpetrator directly. It needs to match someone who shares enough DNA with the perpetrator to indicate a biological relationship. A second cousin, a third cousin, someone whose great-grandparents were the same as the perpetrator's great-grandparents. A family connection that the perpetrator does not know exists in a public database. That match becomes a starting point, and from that starting point, a forensic genealogist builds a family tree backward to the shared ancestor, and then forward again through every branch of that tree until the tree produces a living person who matches the age, the gender, the geography, and the physical description of the person being sought. That candidate then becomes the subject of a discard sample operation. Investigators surveil the candidate. They wait for a discarded biological sample, a coffee cup, a water bottle, a cigarette, a used straw. They collect it without the candidate's knowledge. They test it against the unknown profile. Match confirmed. You have a name. That is how Rex Heuermann was identified. That is how Joseph James DeAngelo, the Golden State Killer, was identified. That is how the Jane Does buried without names were identified. That is how the Darrell Lynn Johnson case was closed 40 years after the crime. And that is the pipeline that is now running the Nancy Guthrie evidence. Let's talk about Othram specifically, because the company is not just a private lab. It is the institution that operationalized IgG for law enforcement in a way that made it deployable at scale. Othram was co-founded by Dr. David Middleman, a computational genomicist who spent years in research genomics before recognizing that the same tools being used to advance precision medicine and ancestry research had a specific and urgent application in forensic casework. The company is based in the Woodlands, Texas, a suburb north of Houston, and it operates as a law enforcement partner rather than as a consumer service. Law enforcement agencies submit evidence to Othram through a formal process. The company extracts DNA using protocols designed for degraded, limited, or complex forensic samples. It sequences the genome using next-generation sequencing platforms. It produces a genotype file from whatever biological material can be recovered, and it provides genealogical analysis and database comparison services to the submitting agency. The company's success rate in previously unsolvable cases is the reason its name keeps appearing in true crime coverage. Not because Othram markets itself to the true crime community, but because the cases it closes generate the kind of public reporting that makes the methodology visible. The Long Island serial killer case is the highest profile example of the standard of evidence the company has demonstrated it can work with. Burlap wrappings, beach exposure, remains that had been outdoors for years. Evidence that standard forensic methods had failed to produce a result from across more than a decade of active investigation. Othram's technology processed whatever biological material was available, produced a genomic profile, uploaded it to the genealogy database, and the family tree pointed to Rex Heuermann. That is the standard. And the evidence in the Nancy Guthrie case, a hair sample from inside the home, a complex DNA mixture with unknown male contributors, evidence that was processed first by a standard private lab that ran it through CODIS and got no hit, is a case for exactly this kind of intervention. Now here's the specific connection to the Nancy Guthrie case, because this is not speculation, this is sourced. When Reuters published its February 12th report about the evidence-rooting decision, Sheriff Nanos sending the evidence to a private Florida lab rather than to the FBI and to Quantico, the Othram CEO, Dr. David Middleman, responded publicly. His word was devastating. Not cautionary, not concerning, devastating. That word came from the head of the company that solved the Long Island serial killer. When the CEO of that company looks at a decision made in a current active case and calls it devastating, he is not speaking generally about the importance of good laboratory practices. He is speaking specifically about what his technology could have done with the evidence that was sent somewhere else. Othram had been positioned as a potential resource in the Nancy Guthrie case from early in the investigation. The FBI's partnership with the forensic genealogy pipeline, which includes formal relationships with companies like Othram that have the NGS sequencing and genealogical comparison capability that standard forensic labs do not, means that once the FBI took effective control of the evidence processing, the pathway to Othram's involvement became clearer. C.C. Moore, who has worked with Othram on cases, described the hair sample as a potential Hail Mary and said the FBI would get another chance with the new evidence. Her framing of getting another chance was not just about a new sample. It was about a new institutional pathway, one in which the evidence is being processed by an institution that has the formal relationship with the forensic genealogy pipeline, including Othram's methodology, rather than by a private Florida contractor running standard STR analysis. The CEO's public statement, Moore's public framing, and the confirmed FBI involvement in the evidence processing all point in the same direction. The specific technology and methodology that closed the Long Island serial killer case is in the lane that the Nancy Guthrie evidence is now traveling. Let's look at the other cases Othram has closed. Because the Long Island serial killer is the most famous, but it is far from the only case where this methodology demonstrated what it can do. The Golden State Killer, Joseph James DeAngelo, 12 murders, 51 rapes, over 40 years of investigation, was identified in 2018 using IgG methodology developed by Barbara Ray Venter. The technological approach was the same. Upload a genomic profile derived from crime scene evidence to a consumer genealogy database. Find a distant biological relative, build the tree, narrow to a candidate, confirm with a discard sample. DeAngelo's profile was matched through a distant relative on GEDmatch. He was identified and arrested within months of the database upload. The Bear Brook case in New Hampshire, 4 unidentified victims found in barrels in the 1980s, 2 of them children, none of them carrying any documentation that allowed standard forensic methods to identify them, was cracked through IgG applied to the victim's own DNA. Othram and the broader IgG community helped identify the victims using the same methodology applied to unknown perpetrators: genomic profiling, database comparison, family tree construction. The victims' identities were established. Their families were notified. Justice for the children buried without names in those barrels became possible because a forensic genealogist built a family tree from a biological sample that decades of standard methods had failed to resolve. The Gypsy Hill murders in San Mateo County, California. 5 women killed in a single year in the 1970s. The case cold for nearly 50 years, were linked to a suspect named Roy Waller through IgG applied to evidence the standard lab had processed but not resolved. Waller was convicted in 2022. His conviction was the end of a 50-year wait for the victims' families. These are not anecdotes. They are the operational track record of a methodology that is now working the Nancy Guthrie evidence. And the specific characteristic they share, cold, unsolvable by standard methods, cracked when the right biological material was processed through the right genomic pipeline, is the same characteristic that the Nancy Guthrie evidence now presents. The evidence was not solvable through standard CODIS analysis. The private Florida lab ran the STR profile. CODIS returned no public match. Standard methods produced their answer, unknown. That answer was not a failure of the science. It was the science accurately reporting that the person who left the evidence was not in the database it was checked against. IgG does not need the perpetrator to be in a database. it needs one of their relatives to be in a database. And the consumer genomics industry has built a database of millions of people who have submitted their DNA for personal reasons that have nothing to do with law enforcement. Somewhere in that database, there is almost certainly a person who shares enough DNA with whoever was inside Nancy Guthrie's home on the night of February 1st to allow a forensic genealogist to build a tree. That tree is what the FBI's forensic genealogy pipeline, the pipeline that includes Otham's methodology, is now trying to build. Now let's talk about the specific challenge the Nancy Guthrie evidence presents to that pipeline. Because the track record is real, but the difficulties are also real. And this investigation's evidence is not the cleanest version of the problem. The complex mixture, Nancy's DNA plus 2 or more unknowns, is the core challenge. In the cases that IGG has closed most cleanly, the evidence produced a single unknown perpetrator profile from a single source sample, a hair from the victim's body, a semen sample, a reference sample from a single biological source. One unknown profile goes into the genealogy database. One family tree is built. One candidate is confirmed. The Nancy Guthrie evidence is a mixture with at least 2 unknown contributors. Before any of that can go into the genealogy database, the mixture has to be deconvoluted, computationally separated into individual contributor profiles. Nancy's known profile is separated from the unknown profiles, and then each unknown profile is treated as a separate genealogy problem. Its own database upload, its own family tree, its own candidate identification process. That is not impossible. The mixture deconvolution software that C.C. Moore described as expedited specifically for this case is exactly the tool designed to do that separation. But it adds a step that single-source cases do not have, and it adds a layer of computational complexity that takes time even when done correctly with the best available resources. There is also the rootless hair issue. We covered this in depth in the last video on this channel. Rootless hair lacks the follicular tag that is the richest source of nuclear DNA. The shaft contains highly degraded short-strand nuclear DNA fragments that fail standard STR analysis, but that can be sequenced using whole genome sequencing methodology derived from ancient DNA research. Otham's technology was specifically developed to address this challenge. Their proprietary extraction and sequencing protocols were designed for exactly the kind of degraded, low-quantity DNA that rootless hair presents. The combination of those two challenges, complex mixture and rootless hair, means the Nancy Guthrie evidence is at the harder end of the spectrum of problems that this technology has solved. It is not the easiest case Othram has ever worked. It may not be harder than the Bear Brook victims, whose remains had been outdoors in barrels for years before the DNA was extracted. It may not be harder than the burlap-wrapped Gilgo Beach evidence from the Long Island serial killer case, where years of environmental exposure had degraded the biological material significantly. But it is hard, and hard means time. And time is what this case has been consuming for 135 days. Here's the thing about the pipeline that people miss, the thing that is easy to skip over when you are reading a headline about IGG or watching a clip about Otham's involvement in a case. The pipeline is not just a laboratory, it is a sequence, a protocol, a chain of specific steps that must be completed in a specific order before the next step can begin. And each step has its own timeline, its own technical requirements, and its own potential failure points. DNA extraction from rootless hair, days to weeks, depending on sample quality and the specific protocols used. Library preparation for next-generation sequencing, days. Whole genome sequencing at sufficient depth to produce a usable genotype, days to weeks, depending on sequencing depth required and instrument availability. Bioinformatic processing, aligning the reads, calling variants, producing the genotype file, days. Genealogy database upload and initial matching, immediate. The database returns matches within hours of upload, but then the human work begins, and the human work is where the time goes. A forensic genealogist, looking at a set of database matches, people who share DNA with the unknown profile at levels consistent with second, third, or fourth cousins, has to build a family tree from those matches backward to a common ancestor, and then forward again through every descendant branch to find living candidates who fit the investigative profile. That work requires genealogical records, birth certificates, death records, census data, marriage licenses, immigration records, newspaper obituaries, Social Security death index entries, local history archives. It requires the specific navigational expertise to distinguish productive branches of the tree from dead dead ends, and it requires the investigative judgment to recognize when a candidate has been identified and the biological confirmation process should begin. In the Golden State Killer case, that tree building process took months. In the Long Island serial killer case, the genealogical work spanned a significant period before Rex Heuermann emerged as the candidate whose discard sample confirmed the identification. The Nancy Guthrie case has now been generating evidence for Quantico since mid-April. The hair DNA data reached the FBI in mid-April. That is approximately 8 weeks as of this video's recording. 8 weeks in the IgG pipeline after the initial laboratory steps are complete is consistent with the tree building phase being active. It is consistent with a forensic genealogist working a set of database matches that have been returned, constructing branches, eliminating dead ends, and narrowing toward a candidate. It is not consistent with a case where nothing has happened. 8 weeks into the tree-building phase of an IgG investigation, with the resources of the FBI's forensic genealogy pipeline and the institutional depth of a company with the track record Othram has demonstrated, is a case where something has happened. The question is how close the tree has come to its terminal branch. Let's bring in the broader context. The context that explains why this specific methodology, applied to this specific case, carries a weight that goes beyond the Nancy Guthrie investigation. The forensic genealogy pipeline has a publicly acknowledged limitation. It works best when the unknown perpetrator has biological relatives in consumer genealogy databases. The coverage of the genealogy databases is not universal. Some populations are heavily represented. Northern European ancestry, for instance, has very high consumer database penetration because products like Ancestry DNA and 23andMe have been heavily marketed in those communities for years. Other populations are less represented. The geographic constraints of the Nancy Guthrie case, Tucson, Arizona, the Catalina foothills, a specific neighborhood in a specific southwestern city, provide a layer of investigative context that supplements the genealogical analysis. The candidate that the tree produces will be evaluated against the geographic and demographic profile of the case. A tree that generates a candidate family concentrated in the Pacific Northwest is less compelling than one concentrated in southern Arizona or adjacent areas. The physical description from the surveillance footage adds another layer. An unknown male, approximately 5'9" to 5'10", average build, present in Tucson on the night of February 1st. The phenotypic predictions that the FBI's NGS data can produce, probabilistic physical appearance estimates derived from the genomic profile itself, provide a biological description that can be compared against the genealogical candidate. Those layers, forensic genealogy, geographic context, physical description from footage, phenotypic prediction from DNA, converge on a single investigative question. Does the person who fits all of those profiles exist as a living candidate at the end of the family tree that is currently being built? That question is the one the Othram caliber pipeline is designed to answer. And the answer, when it comes, will not be a probabilistic estimate. It will be a biological confirmation from a discard sample. It will be the same kind of clean, court-admissible result that sent Rex Heuermann to a courtroom after the Long Island serial killer investigation had been open for 13 years. Here's what the Long Island serial killer case tells us about the specific moment the Nancy Guthrie investigation is in right now. Because the parallel is not just technological, it is temporal. The Lisk case was officially cold, not abandoned, but cold, for more than a decade before Rex Heuermann was arrested. The forensic genealogy work that identified him did not happen in a single season. It happened across a sustained investigative effort that required methodological evolution, database growth, institutional partnerships, and the specific deployment of technology that did not exist in 2010 when the bodies were first found. The Nancy Guthrie case has been open for 135 days. 135 days is not 10 years. But those 135 days include a period in which the most powerful forensic tools available to American law enforcement were not being applied to the evidence, because the evidence was in a private lab in Florida, not at Quantico, not in the IgG pipeline, not in the hands of the institution that has the technology, the relationship, and the track record. The effective clock on the forensic genealogy investigation did not start ticking on February 2nd. It started ticking in mid-April when the FBI confirmed receiving the hair DNA data. 8 weeks ago. 8 weeks of tree building. 8 weeks of genealogical navigation. 8 weeks of the specific kind of quiet, labor-intensive, historically sourced investigative work that produced a name in the Long Island serial killer case, in the Golden State Killer case, in the Darrell Lynn Johnson case, and in hundreds of other investigations that ended when the family tree found the right terminal branch. The investigation that caught the Long Island serial killer is running Nancy Guthrie's case. Not metaphorically, not aspirationally. The specific technology, the specific institutional pipeline, the specific forensic genealogy methodology that identified a Manhattan architect as one of the most prolific serial killers in the history of Long Island is the same methodology that is working the hair sample from a house in the Catalina foothills of Tucson, Arizona. The lab that caught Rex Heuermann is looking for whoever walked through Nancy Guthrie's front door. And somewhere in the genealogy database, in the profile of a second cousin or a third cousin who took a consumer ancestry test for reasons that had nothing to do with a February night in Tucson, there is a branch of a family tree that ends with a name. The genealogist building that tree does not know yet if they have found the right branch. Or maybe they do. Maybe the branch has been found. Maybe the discard sample is already being arranged. Maybe the name already exists in a case file that has not been made public yet. That is what the silence sounds like from the outside. It sounds like nothing is happening. It sounds like the investigation has stalled. It sounds like 135 days of unanswered questions with no resolution in sight. But from the inside of a forensic genealogy investigation at this stage, from the inside of a pipeline that has closed the cases that nobody else could close, the silence sounds different. The silence of a family tree being built sounds exactly like this. Quiet, specific, relentless. The same way a centrifuge sounds when it is spinning.